So thanks for the invitation, and it's a great pleasure to show you some of the work that we actually developing in my lab in Mexico at the moment. So we are actually a great talk from Cesar yesterday and Eva today, talking about the application of AI toward peptide design and especially machine learning models. Today I'll be talking mostly about the limits of the current models and how we can improve them. So let's see, that works. So as we know, I mean, I'm going to go quickly on that, but basically peptide design, protein engineering is basically the idea of altering a protein sequence or potential structure in order to modify and understand better the current properties and possibly improve their functions. And the traditional way I've done it, it seems that Joe has done it as well, doing the mutation, whether it's truncation, alanine scan, or a specific mutation, you end up doing a lot of mutation to understand better the role of each amino acid within the function of the structure. It's also, when we look at this way, we tend to assume that the structure remains the same when it's not necessarily the case. One of the challenges also is to be able to navigate the peptide space, which are relatively large. So if you were, I think Cesar presented something similar yesterday, when you imagine the whole peptide space as this black rectangle, and then you imagine that your peptides represent only a small niche, a small side over there, and all the analogs around it represent all these little mountains. So there will be mutation on a specific scaffold, and the right key, the right mutation will lead to the top of these little mountains. So you will end up having to explore for that, like a large number of sequence space, eventually hit and being lucky, or you can just drop and not having any biological function for breaking the bonds necessary to the maintain of the structure or the function. And so at the end of the day, basically the peptide sequence structure and functions relation to that are relatively complex, but we got limited time, limited resources, and not necessarily the fundings to actually doing endless research in that area. Therefore comes computational protein design. So here you start basically from a seed protein or proteins or peptides that you actually Eva talked about embeddings earlier. So the idea is to be able to make the information you find relevant to the model necessary to be understood by the computer. And then what will substitute the synthesis, it will be the generative modeling, whether it's an alanine scan, whether you want to do maintain a specific key structure or key elements, dimer, trimer within an actual sequence. You can do deep mutational scanning, or you can do analogs, etc. And there is a lot of generative models that are coming up these days to make all these sort of, depending on the question you ask. On the other hand, you will test all these sequences over most of the predictive models. It's true that most of the models today are built on sequence-based and not necessarily contain structural information. And at the end, so that's very much similar to what you will do in synthesis. Traditionally, you start from one ligand, you'll do some mutation, you'll test the biological activity, and eventually you choose one or more biological activity, whether it's a KI, an IC50, etc., and then to basically filter out and find the optimal sequence, the desired one. The difference is maybe in the way you explore the sequence space. When you do sequence on solid phase peptide synthesis, you will do certain rationals, doing small step by small step, just to make sure that the story will be right for your paper, and that will be ideal easier to explain to the reader. When it comes to computational peptide, you explore a large, wider, and not necessarily related sequences, and then you follow eventually the fitness function or more than one fitness function to actually tune and optimize the sequences. Actually, one of the challenges, we often talk about the machine learning as if we do one model at a time, but the reality is that when we do peptide design, it's basically a multiparametric optimization problem. You don't have one, but you have many functions for the same sequence space. You basically can explore the high toxicity, the specificity versus immunogenicity, broad spectrum. The thermal and pH stability is one of the other problems, proteolytic degradation, or membrane permeability or oral bioavailability, one of things. So you can imagine that here is you can explore a given sequence space and then apply those multiple predictive models to that same sequence space in order to identify those same different importance. So we did that actually. Initially, we looked at develop some of the early 2021, 2020 hemolytic peptide models along some other researcher. And what we were also interested was to actually understand what was the limit of those models. So what we applied was to, on one side, the predictive models. So what you see on the top is basically with the orange to purple. So it's basically gradient boosting classifier that we applied to our training set on one side and a test set on the other. And basically the shade of purple will lead you to an hemolytic structure. What you see on the bottom is we were interested to understand, based on what we know from the training set, what the model cannot predict correctly. And so that means what structure or sequences will be outside the scope or the domain of applicability of our training set. So we apply multivariate outlier detection model. So you can buy a univariate that should allow Nebis distance, but we were interested to apply a multivariate approach. And so one of our models was based on average KNN, and works on all three sets of, we had three training sets, and they were, the method worked on all three of them. So what you see at the bottom, basically the darker is the red or the darker is the blue, the sequence is outside the scope of prediction of the actual training set. So that means we can actually, you cannot basically rely on the predictions that your original model gave. We actually decided to do a de novo design of random peptides to emphasize those trends. And we first generated de novo 5,000 sequences that share some amino acid distribution with AntiMicro Peptides. So what we noticed is that in that example on the left side, that sequences that were rich in small residue, in particular glycine and proline, were likely to show high outlier score, meaning that those sequences are often not, those residues are not often represented in a training set. Likewise, when we did the same analysis on the same data set, but this time on comparing the negatively charged versus positively charged residue and the density, we saw that basically many cationic peptides often associated with a high probability of being hemolytic in that case. So that's one example when basically the sequential information also matters when it comes to understand what are the limits of your peptide. You can have a sequence, but not all the peptides will be necessarily predicted correctly. We actually applied these kind of models to a large number of antimicrobial families. So here is the idea to apply machine learning directed evolution to design specific non-hemolytic antimicrobial families. So in that previous paper that I was talking about, we estimated about 70% of the antimicrobial peptides were considered hemolytic or hemotoxic. And so the idea is to explore with potential a minimum number of mutations, mutate some of those families, we can eventually design novel antimicrobial peptide with a reduced hemotoxicity. And so we're still exploring this because there's about 267 families, and basically the idea is to collaborate with many peptide scientists that are dedicated to specific families, such as David Craig in Australia working on cyclotides, or Mariela Mariani in Argentina working in amphibian antimicrobial peptides. And so the idea is to be able to, so we get the very first one called OrthoMCL0. It's actually a family of brevenines, which is the most documented within this data set. And what we actually plot here is the helicity on one side, because many of those antimicrobial peptides turned out to be helical. And we also interested to measure the Jensen-Shannon distance, not because just because we are interested in terms of conservation of the sequence and be able to target the sequences that are the most conserved first, so we have less mutation to do. The other sequences will not, amino acids will not necessarily be associated with the biological activity. Another example of that application is when we actually applied those to multiple predictive models. So Christina, during the master, basically developed, we're looking for sequences that had non-hemolytic anti-cancer activity, and specifically with selectivity towards specific cancer cell lines. So she started with 140,000 peptides, and then applied a succession of binary classifiers, so anti-cancer, non-anti-cancer, hemolytic, non-hemolytic, selective, non-selective, to end up with a handful of peptides that we currently are currently synthesizing with other Australians in the lab of David Fairley and test against different cancer cell lines over there. The second example of potential importance of being careful in machine learning design models is, for example, many of those models have been applied to AntiMicro Peptides. And what's actually interesting is that here is an example of the 3,200 AntiMicro Peptide natural that have been reported in the APD database. It turns out that 75% of those peptides do not have a 3D structure, and 15% of the 24 remaining actually consider helical. So the perception that most antimicrobial peptides will be helical commonly come from what has been published as PDB structure. And so the perception is basically the structural information is largely unknown, and we may eventually have an amino acid composition and derived property that will favor alpha helices. So we were actually wondering whether within the database that we're using, because most of the machine learning algorithm that we use are entirely based on sequences, and then we don't know the structure in the first place, unless you apply AlphaFold or anything to your entire data sets. You basically will likely to have in those training sets basically helical structure, mostly because it largely represented. And eventually, these will actually bias to the generation and the prediction of antimicrobial peptide, limiting basically the robust exploration of other possible sequences. So what we did with Daniel was to benchmark, and that was in the pre-AlphaFold era, benchmarked because Rosetta was still 3 days. It was taking several days to predict the secondary structure. Now Rosetta Alpha is shorter. But basically what we were doing was to explore different secondary structure predictions methods in order to understand what will be our possible structural landscape of the largest repository of antimicrobial peptides. And so we evaluated 3 that actually can handle large, and are actually continued because many of those tools are actually not maintained. And we used adjacent divergence in order to understand whether between the 261 sequence and structure pair, we had a lot of deviation or not, and if basically the prediction we had between the structure from PDB and the one from predicted from those methods, I mean, we will end up having a small deviation for it. So here's an example when we can actually display this structural space. We use these ternary plots when we use the helix, coil, and sheets percentage, and you can eventually see basically the distribution of 261 analogs peptides with the different folds they could adopt. And I shows here 6 examples when you see that eventually the true what we consider the ground truth with the PDB structure and the predicted structure, let's say the predicted coordinates, are usually close to one another. And so that allowed us to actually map GRAMPA. We're able to show that basically GRAMPA is largely biased. So that was what we were eventually suspected, but because no one had basically a 3D structure of the entire database, so it was actually interested to explore. So what we see is that mostly 30% if not more, 50% of the actual grandpa data sets are mostly folded within helical and coil structure. And the rest will be adopting mostly coil, beta sheets, and even a mixture of the three alongside the thing. So another, when we look at a subset specifically towards the one strain, say E. coli, so we show that we lose some of those non-helical structure. And if we look, for example, the example of E. coli, the data set that was used for the publication of that paper, Peptide, the authors actually suppressed or removed the peptide, the sequence that were rich in cysteine and prolines, and that led to restrictions of the space towards mostly alpha helices and coil and a mixture of the two. And without surprise, basically, the peptides that were synthesized were mostly alpha helical and coil. So what we did after that was to actually evaluated whether these sampling bias were eventually affecting the predictive models in place. So we evaluated 13 of those. And so we evaluated, for example, here you use grandpa, and we also set up another data set called non-grandpa. And here we're just looking at just the region of grandpa and non-grandpa that actually folded into an alpha-alpha coil, so that's small. So we see that most of the models are actually predicting relatively well when it comes to the alpha helical structure, and with few errors. But when it comes to beta coil and mixed structure, then there is a problem. Then we see that some are completely misclassified, and we ended up having a lot of misclassifications, which is one of the issues. And most of this misclassification has to do with the fact that the structural space associated with the training set does not necessarily overlap with the one with the test set. And in that particular case, the one represented by the negative sets of each of those models basically are deprived of beta reach or beta sheets reach or mixed structure, which led to a lot of misclassifications. And with that, so basically the general conclusions is that predictive models can be used as surrogate for biological testing. It's quite necessary to do a systematic use of outlier detection or uncertainty approach in order to understand if we can actually use one or more machine learning, deep learning models upon a sequence space. So that means if you were to explore a peptide for AntiMicro Peptide immunogenicity, well, it'd be good if the two spaces overlap. I mean, if they are alpha helical, because most of them mostly will be alpha helical, that's kind of the safe area. Also, if you were to look at other, maybe more complex or more original structure, that'll be harder to make sure that they actually overlap in that case. So eventually you will be using generative models to compensate or try to get more generalized model. We also use it for explorations. And so, like I said, structural bias in peptide modeling is real. And we see that actually right now for AntiMicro Peptides, they mostly gave favor towards alpha coil AntiMicro Peptide, meaning that many structures that are not in this area will be misclassified in general. And that's my group. So you just listen a couple of results from two of my students. And here's a different area my lab is working at the moment.